Hi, I'm Richard Sever from Cold Spring Harbor Laboratory Press. With me I have Ron Evans from the Salk Institute. Ron, welcome to Cold Spring Harbor.
Thanks, Richard.
Now, you told a fascinating story last night about vitamin D and the connection with pancreatic cancer. Now, what I'd like you to do first is if you could just paint the broader picture. The vitamin D receptor is a member of the family of nuclear receptors that you discovered 30 years ago. So can you just tell us in general terms about nuclear receptors and what distinguishes them from the kind of cell surface receptors that people may be more familiar with?
Well, it's a good question. Nuclear receptors, of course, by their name, reside in the nucleus. That is, they exert their effect by interacting and controlling gene networks. And the particular feature about any one receptor is that its duty is to find specific sites in chromatin and either unlock those sites or in some cases to actually repress the sites. And so they can be both activators and repressors. And so the way to control something very complicated like a physiologic response that involves many genes is to literally marshal hundreds of genes to be activated and hundreds to be repressed. And receptors respond to ligands, and therefore you can have this highly regulated network that is tuned to specific physiologic challenges, either high-fat diet or starvation or changes in cholesterol. And so many aspects of our body physiology are controlled by changing gene activity in the nucleus as opposed to the way other receptors work, which is at the cell surface. And so it's a fundamentally different type of signal transduction, but it's very effective and serves a specific role that is different than the cell surface type.
And so what distinguishes the ligands for this type of receptor compared with other receptors?
The nature of the nuclear receptor signal is typically a chemical, small molecule that is more often than not a metabolite that is part of the pathway that the receptor is trying to control. So, for example, bile acids can be involved in digestion, but they're also a functional ligand for nuclear receptors. And so as a result, the receptors that respond to bile acids control bile acid metabolism. Mm-hmm. And most of the ligands for nuclear receptors are lipids, so they can get through the lipid bilayer. So fatty acids would be typical ligands, as well as cholesterol derivatives would be typical ligands. And so lipids, cholesterol, they get into the cell and then control pathways that control the actual metabolism of those molecules themselves.
And so what happens with steroids? Because they're also ligands for some nuclear receptors.
So steroids are modified forms of cholesterol in particular ways just to allow them to be a little bit more soluble in solutions. So cholesterol is very hydrophobic, and the steroids are slightly chemically modified to be able to interact with receptors. And so steroids, again, control their own production and also their release from organs such as the adrenals or gonads is typically controlled by either metabolic stress or neuronal type of signaling. So neurons also control the release of hormones from glands like the adrenal gland. And so there's a network of hormones that are circulating through our blood. Basically, because of their chemical nature, they're permeant to every tissue, including the brain. So no tissue is free from being fully exposed to steroids. And as a result, they can control neuronal activity, visceral activity simultaneously and coordinate what we'd call the physiologic response.
And so you would see that as being distinct from many of the other ligands for cell surface receptors in terms of that high-level coordination that's hitting like every cell, and there's a level of combinatorial action that is a more higher order. Would you say? Is it safe to say?
Well, I'd say it's a different word. It's a molecularly distinct type of signaling pathway. It does not involve the typical bind to a cell surface, activate a kinase on the intracellular side, and then use a series of cascades to transmit an effect within the cell. The logic of the nuclear receptor is to directly have the ligand on the gene that is being regulated itself, or the genes, I should say, because it's a network of genes. And the genes are going to be different in every cell type. And so if there are 200 cell types in the body, there would be 200 gene expression programs that are simultaneously being activated. And that controls and coordinates this physiologic response. And so it's using transcription as the core underpinning of the physiologic mechanism.
And so in this control of transcription, as I understand it, each nuclear receptor has two regions, the DNA binding region and the ligand binding region. And one of the points you made in your talk last night was that these are not separated. In many other signaling molecules, you see this kind of domain shuffling during evolution where different modules are paired with other modules. And it seemed like that doesn't happen with nuclear receptors. Why is that?
That's a fascinating question, and one of the points I made is that the nuclear receptor motifs of the DNA binding domain and the ligand binding domain are unique motifs. That is, they're not seen outside of the animal lineage. So it's a metazoan-specific motif, and there are only two transcription factor families that were not invented by bacteria or yeast. Nuclear receptors are one, and ETS transcription factors are the other. And so animals have something unique. We do not know the origin of unique genes, but unique genes have unique motifs, and the unique motif therefore gives the potential for something really new. And something that's new in animals is physiology. So we think the origin of the physiologic mechanism resides in the novel motif of the receptor itself. One is the unique DNA binding domain. The other is its associated ligand binding domain that allows it to sense chemicals and to use chemical signaling to actually control transcription. And so this became a new way of signaling. It's common in all animals. We don't know any exception, but it's not present in plants or any other type of organism.
Right, and one of these chemicals that is sensed is vitamin D, and the story you told last night was that vitamin D receptors start showing up in injured liver and pancreatic tissue. Is that correct?
That's right. So vitamin D it was isolated back in 1917. It was the fourth vital factor. So the first one was vitamin A, then there was B, C, and then D. And so it is 100 years old. But actually how it works is just only being deciphered. And it principally works through the vitamin D receptor that binds to a very specific nucleotide sequence. And we thought we knew what it does, which was control calcium, very important process in bone mineralization and bone remodeling. But what we discovered is during injury, while the normal liver has no real levels of the vitamin D receptor, the injured liver has a very high level. And that high level expression comes from a particular cell that's involved in wound healing. And so that cell expands during the wound healing process or the injury process, takes care of the wound normally, and then regresses back to its quiescent state. And this process that leads to tissue fibrosis and wound healing, their linked response, normally maintains tissue function, but under chronic injury, such as a viral infection, or a parasitic infection or a cancer, that acute response cannot turn off, and you get chronic wound healing. It's like a wound that won't heal. Continual inflammation, continual fibrotic response, and then the liver ultimately progresses to liver cancer, and the same process happens in the pancreas, and that triggers the progression of pancreatic cancer. And so we're interested in the fibrotic response as a wound healing mechanism, but also as it encourages cancer. And what we showed, to be brief, is that this response, sometimes is called the stromal response. It's the surrounding cells, which are not the cancer, but they're trying to fight off the cancer, actually encourage the cancer. They provide sort of the fuel supply line. And that fuel supply line is kind of like a living skin around the tumor. If you give a vitamin D ligand, it will turn off that chronic response. The skin will open up, drugs can then rush in, and also the T cell response, that is autoimmune type of response, can now flood in and immunologically try to remove the tumor, as well as let drugs try to kill the tumor. And so we're having great success actually in using a simple molecule, a vitamin D ligand, to open up a new type of therapeutic approach to treating one of the worst cancers ever, pancreatic cancer. And so, deceptively simple. The science is actually interestingly complex, but the therapeutic approach becomes relatively easy. So,
Right.
it's one of these wonderful surprises. We're very excited by the discovery and its rapid therapeutic potential, that we're now taking this into clinical trials, which have already started. And so patients, as we sit here right now in this seat, are being treated at a number of different hospitals and beginning to let us evaluate the therapy, which is looking already very promising.
So in terms of this notion that cancer is a wound that doesn't heal, the vitamin D receptor, what's that doing in a normal cell that isn't happening in the cancer cell? When's the vitamin D receptor showing up, and what's its role that doesn't seem to be happening in cancer, or is it the vitamin D ligand that's missing?
Well, there's two things happen. Under damage, tissue damage in the pancreas or the liver, the wound healing response gets activated. Normally that's a short process because it's mechanical damage. It can be repaired, cleared out, and then when the damage signal goes away, which is a molecule called TGF-beta, then the whole system quiets down and returns to a non-active state, so-called quiescence. But with cancer, the tumor is stimulating this process, and so the wound healing response has no mechanism to turn off. So we go in and turn off.
So vitamin D can be considered the off switch in
It's
this process?
it's the brake. It's kind of like
Right.
a molecular brake.
Uh-huh.
And the ligand, vitamin D itself, is not present in the environment of the tumor, and so we supply that with a drug. It's either an injectable form of vitamin D, a potent form, or a pill.
Uh-huh.
Specialized form that is much more potent than the one that you would take as a supplement. So it's hard to use the supplement to.
So you can't just take large doses of vitamin D and hope.
You could, if you got a lot of sun exposure, because taking a supplement is just the beginning of getting the active molecule, and you need good sun exposure. And so if you're very disciplined, you can do that, but you have to be careful because the supplement can also give rise to high calcium, which you don't want to raise that too much. Now, if you have pancreatic cancer, you may not care.
Right, right.
But the pill form is actually relatively inexpensive, it's much safer, and it's more effective. So if there's an option I would encourage that. And I have to say, it's not approved. We're just at the beginning of the road. Typically, cancer therapy is a long road to get to FDA approval, but because we are using an already approved drug, we're hoping it's a short road, especially because the disease is so devastating. We may literally be offering a type of new therapy that's very powerful, but also very simple. And so if we can do that, we'd like to get that to the patients quickly.
And in terms of dissecting at the molecular level how this treatment is working, how far are you along with that? I mean, you mentioned last night that signal seems to essentially antagonize signaling via interleukin-6. Is that correct? How are you picking apart those pathways?
The activated cells produce an impressive number of collagen molecules. 18 different collagens alone are being deposited in the environment of the tumor. T cell blockade molecules like CXCL12 that prevent the immune system from coming in and invading the tumor, and powerful cytokines like IL-6 that are often linked with cancers that are in their very active stage of expansion. And so vitamin D treatment very effectively and relatively rapidly turns off all those signals, literally hundreds of genes, but all the collagen genes, these T cell blockade genes go off, and IL-6 goes off. And each one of those by themselves would be a benefit, but turning them all off, you're getting a multi-pathway effect by giving just a simple chemical. And that's the advantage of targeting a nuclear receptor, is you hit hundreds of genes,
Right.
as opposed to just using, there's an antibody to IL-6, you would just take out that one component.
Right.
There's an antibody to CXC12, you take out that one component. But we're taking out those components and hundreds more that are complementing it. And so we turn off the entire process for a window of time. And the window of time allows drug to come in and immune cells to come in, and that's the advantage. It's not actually killing the tumor. What we're allowing is the tumor to be killed.
Right.
And so we depend on the body and drugs.
So in the idea within pancreatic cancer is to use it as a combinatorial therapy along with
Exactly.
treatments.
So we believe that without the other components, without the immune component and the chemotherapy, it would be much less effective. So in the combinatorial setting, which are already approved drugs, but they're very ineffective, the effectiveness of these drugs goes dramatically higher with the vitamin D. So it's a synergizer. It is a simple molecule that really enhances the effective therapy to a new level.
So, I mean, thinking of the piles of vitamins that various people take every morning before their breakfast, is this one of these ones that we should actually be taking?
Well, most people, the average person, is not vitamin D deficient. On the other hand, most people who are sick are vitamin D deficient. So chronic illness leads to a progressive lowering of vitamin D levels in the body. And with that reduction increases your risk for something happening somewhere. And we believe that the lower level of vitamin D that's seen in many studies for many diseases is the reflection of this wound healing response that consumes the body's vitamin D supplies. And when they are consumed, it's hard for that response to turn off. And so by resupplying it with our supplement, I won't really call it a supplement, it is a drug called Zemplar, that's what we use, or paracalcitol. That is either injected or taken by a pill, and that goes directly to the sites of deficiency and reverses this process. And so it's actually a little bit more complex than I was alluding to, but the way in which the physiologic response works, all of the vitamin D that we're returning to the body goes to the right spot almost immediately. It goes to where the low levels are lowest. and fills up that bucket first.
Right.
And as a result, you get a very quick response, literally within days. And so for patients, that's really good news. We think it's going to be a huge benefit because the currently approved drugs are very ineffective, but we think with this, they'll be highly effective.
Right. And I mean, so pancreatic cancer, then that must be, is that just the first step? I mean, one can think of there's many cancers, many wounds that don't heal. There's, as you talked about, fibrosis, various other conditions that are chronic. Are you looking to other conditions to follow the work on?
Well, it's our view that this is the start, and every new kind of therapy needs a start. But most cancer drugs find uses in other cancers. And so this is one big step forward, but we think there are going to be many steps that will apply to other cancers. So our next ones that we're looking at would be kidney, liver, colon, lung, breast. And they all have a fibrotic response. There's no literature that says it's the same kind of response that we see, but we believe that in fact it is. And so once we tie up those loose ends, then, and if we're correct, then vitamin D should be very effective in the same way as improving the therapy in these other diseases.
Well, it sounds like it's the one big step, the first of many. It's been very nice talking to you, Ron. Thanks very much.
My pleasure. Thank you.